Hi guys, and welcome to Activity 4 for Biological Sciences Year 10. In this unit what we're going to be looking at is sex and chromosomes. Basically, how do the chromosomes determine what sex you're likely to be? Obviously whether you're going to be a male, whether you're going to be a female. Okay, now the first thing to understand is that we've got, in our 23 pairs of chromosomes in the human genome, basically what we've got is one set of pairs which we classify as the sex chromosomes. They're the ones which are going to determine whether you are going to be a male, whether you're going to be a female. Now, when they're all put together, when scientists look at the various types of genes, they've classified these as the 23rd pair. Okay, why? Well, basically it just makes life easier for us to be able to identify the sets of pairs, and then as our information gets better at understanding what makes up each of our chromosome pairs, we can then say, well, on chromosome pair number 1, we've got this type of information. Chromosome pair number 2, we've got this, etc., etc. So the 23rd pair basically corresponds to your sex-determining chromosomes. Now, something very unusual about the sex chromosomes, and the unusual thing is that in all the other cases where we've got our chromosomes, the pairs are the same size. Okay, so we've got duplicate pairs from mum and dad. They're going to be exactly the same size. In the sex chromosomes, we've got some which aren't. Okay, now they've been identified as having the letters X and Y. Now, X is basically a large chromosome, and Y is a diminished chromosome. Now, by putting these two chromosomes together, basically what we get is a variety. Now, the way in which we look at it is that the Y chromosome, because it's smaller, basically is deficient in a specific number of genes. And we'll talk about that later on, right at the very end of the genetics unit when we look at sex-linked diseases. But basically what you need to understand is it's like having a chromosome that's been chopped off but it survives for some reason. And by putting an X and a Y together, you'll get one type of individual. However, if you get an X and an X, you'll get a different type of individual. Now, females basically are made up of individuals which have two X's. Both chromosomes have a full complement of genes, whereas males have an XY. They have one chromosome which has a full complement of genes, but they've got a Y chromosome which is deficient in a certain number of genes. So as a result, what that means is if you're deficient in a certain number of genes, whatever is in the full complement chromosome will code for your variety. Now, if that was coding for a disease, then you're going to get the disease. That's why you find that some males may have certain diseases like colour blindness, haemophilia, and sickle cell anemia is another one. And basically what you tend to find is it's because that the X chromosome, which has got a full complement, its gene basically codes for a disease, and there's nothing to back it up from the Y. So the result is it gets coded. So let's just recap. In order to get a female, we're going to have an X and an X, two big chromosomes, which means that we get an X from the mum, X from the dad, come together, and they battle it out for what gene is going to be the dominant one. In a male, though, what we get is an X chromosome and a Y chromosome. You're going to get the X from mum, and you're always going to get the Y from dad. There is no YY. Okay, that's not possible. So you'll get the X from mum, and dad will give over a Y. So you'll have a sperm which comes along and basically fuses with the egg, and that sperm will have a Y chromosome in its 23rd position. Okay? The X will come from mum because all of her eggs have got XX. So that basically determines what a female is. XX is a female, XY is a male. And remember that the gametes only contain one chromosome. So they don't have a pair, they just have one chromosome. Alrighty, so let's look at how we determine whether you're going to get a male or a female, and what's the probability of getting that type of offspring. Well, what we need to do is, and this is what scientists do all the time, what geneticists do all the time, is they always work out the probability. So we do what we call as a genetic cross. Now, genetic cross basically looks at what gametes have we got from the male, what gametes have we got from the female, what's in their nucleus, what have they got. And when we put them together, we do it for a single gene, a single trait. We put them together, we say, well, that could join with that, or that could join with that, that could join with that, that could join with that, and as a result, we get this and this, or this and this, and that way we work out a probability. So let's look at the probability of getting a male. Now, obviously, in order to get an offspring, you must have a male and you must have a female. We know that in the gametes, remember the gametes are going to have half the information because of meiosis. In the gametes, we're going to have X and Y for the male and X for the female. We have no idea which one's going to fertilize. So this is how we do the cross. There's your parents. There's your gametes, XY for the male, XX for the female. Then we produce something called a Punnett Square. Now in that Punnett Square, we're going to put our information. We've got our male and our female. Okay? Then we need to put in our gametes. Now the gametes go in each individual box. So basically in the first box, that's a sperm which contains an X chromosome. Now half the number of sperms will have that X chromosome. In the other half of sperms, they're going to be Y chromosomes. In the female, half the eggs are going to have that X chromosome and half the eggs are going to have that chromosome. Now notice that the X's are all different colours. This is going to allow us to have variety. But the key to understand is the size is the same. Okay? Because the size is the same, we've got the genes in exactly the same place. So what happens? Well, let's say that the red sperm fertilises the orange egg. Well, what we'll get is an XX individual. Now, another probability that we could have the red sperm fertilising an ovum which has the blue type of the X chromosome, but they're still XX, they're still female. So both of those are going to be female. Or we could have the yellow ovum from the egg, the yellow X, joining with a Y chromosome, a green Y chromosome coming from the male. Then we get an XY, orange, green. Or we could have an blue X chromosome from the mum, which is fertilised by a sperm carrying the Y chromosome, which again is green. So we get another male. Now, if you look at the colours, you've got red and orange, red and blue. Those are the probabilities that you get, but both of those are female. And if you look on the second column, you've got orange and green, and you've got blue and green, but they're both XY, XY, so they're both males. So the probability of getting male or female is 50%. And it's always going to be 50%. Every single time the individuals reproduce, the chance of getting a male or female will always be 50%. So this is a good example of a genetic cross where we're actually using the idea of X and Y chromosomes as being our genetic traits. Okay? So I hope you found that useful. Hopefully you've done this in class, so this is more of a revision process, and as we go on you will find these genetic crosses getting more easier. Now, I have put a couple of slides together that you can practice, but also I just wanted to show you and link you back to the app where basically we have, they showed a really good carrier type. So I've taken a screenshot of the app. Now this is in that very first section, genes and inheritance. So if you go through and watch that, you'll find that useful. And you can see down the bottom, you've got the 23rd chromosome, and you can see we've either got the female, XX, or the male, XY, in that red circle. Remember that red circle's not there, I just put that there to identify it. And you can see the 23 pairs. And basically, by putting this together, this carrier type lets geneticists be able to identify the various chromosomes and any problems. But notice the sizes. They're all identical in sizes, which means one came from the mum, one came from the dad. Only the 23rd chromosome changes. Okay, well I hope you found that useful. Go on using the activity and have a go at exercise 4B. And yeah, join me again. I hope you found it informative. And by all means, if you have any questions, go and ask your teacher or come back, re-watch the video. And I hope I've explained it well enough that you'll find it useful. Thanks for watching.